Hello, welcome to this session on protein synthesis. My name is Winnie Barawa. In this session, we are going to explore how the proteins are made all the way from the DNA until we end up with the chain of polypeptides or amino acids. So if you're new here, this is a channel that focuses on lectures, lessons, and also expert interviews in the arena of healthcare. Don't forget to subscribe if you've not done that and be with me all the way to the end. So protein synthesis, this is the process that helps us in the formation of proteins, and in some places they prefer calling it protein biosynthesis, still the same. However, protein synthesis is divided broadly into two major phases, and this includes the phase of transcription and the phase of translation. So in the transcription phase of protein synthesis, this is the process where the DNA will encode a protein known as a gene, and it's converted into a template molecule called the messenger RNA. So in short, transcription is the process of creating a, a molecule or a strand of an RNA, which will be used now in the translation stage. So in the translation stage, the messenger RNA that was formed in the transcription process will be taken to the ribosome where it will be read by these ribosomes and create the different sequence of amino acids that will together give us our DNA. So in summary, how does protein synthesis happen? The protein synthesis originates from the DNA, where the DNA will go through transcription, and that is formation of a messenger RNA strand. And this messenger RNA strand will therefore be taken to the ribosome, where it will be translated by the ribosomal RNA and help us in forming the different amino acids that will be attached to one another to give us the polypeptide strands or the polypeptide chains. So that's a summarized way of protein synthesis. Happens with two main stages, transcription and so let's start with the transcription stage of protein synthesis. Transcription is the process of transferring genetic instructions in the DNA to the messenger RNA. And the transcription process happens in the nucleus of the cell. So what happens in this process is that during the transcription, a strand of messenger RNA is made from the complementary strand of a DNA, what we call the gene. So the transcription takes place in three steps, whereby we have the initiation stage, elongation step, and the termination. So the transcription in itself happens in three stages. There's initiation, elongation, and termination. Initiation simply means starting of the transcription stage. Elongation means elongating the chain, that is the RNA chain. And termination simply means stopping this process so that we can progress to the next stages. So in the initiation stage, this is the stage whereby we have the enzyme known as RNA polymerase that will bind to a region of a gene that we call a promoter. A promoter is like the primer during the DNA replication. So what the promoter will do, it will signal our DNA to unwind, okay? And the RNA polymerase enzyme can now read the bases in one of the DNA strand. So here we are not making two complementary copies of the DNA. We are making a simple complementary copy of an RNA from our DNA strand. So the enzyme, that is DNA polymerase, will read the strand of the DNA and it will make a messenger RNA strand with the complementary required bases. Whereby, because we are making an RNA strand, what happens is in the DNA, we will not attach or we will not pair adenine to thymine. In the space where we're supposed to pair thymine, we shall replace it with uracil. Okay, but in the case where we have cytosine, we will correctly pair it guanine, and in the case where we have guanine, we shall correctly pair it with the cytosine. So that's how the initiation starts. We have the RNA polymerase enzyme that will bind to the promoter, and the promoter will signal our DNA now to unwind and allow the RNA polymerase to read our bases in the DNA strands to start forming the messenger RNA strand. Keeping on with the transcription, the next that follows is the elongation step. So in the elongation step, this is basically progressing with the messenger RNA strand by adding more nucleotides in the required base pairing, such that where we are saying where we have a cytosine, we will pair it with guanine. Where we have an adenine, we shall pair it with uracil instead of thymine, because in the formation of RNA, we shall replace thymine with uracil. And at the termination stage of our transcription, this is the ending of the transcription process, and the RNA polymerase transcribe a terminator. So a terminator is the stop code that will allow the DNA to detach, or it will allow the RNA to detach from the DNA. And at this stage, the messenger RNA strand is complete. And after this stage, it can go to the next stage of the transcription. However, we are being told that before the messenger RNA can be used to be processed now in the translation process, what happened is we need to edit this messenger RNA to remove any damaged sections to ensure that it is complete before it can be processed into amino acids. So the transcription process has initiation, where we start with the beginning of the promoter and the DNA unwinds, and the next stage is elongation, where we shall create the strand of messenger RNA that will be used for our amino acids processes. And the last bit is termination, where we have a terminator that allows the process to stop and the messenger RNA to detach from our DNA and be taken to the cytoplasm towards the ribosome for the process of forming the amino acids. So going forward, this focuses on the editing of the messenger RNA, and we shall just call it processing of the messenger RNA. So we are explaining that in the nucleus is where the transcription happens, and in the transcription process, we shall form a copy of messenger RNA. And the messenger RNA, before it can go into the ribosome for translation, it needs to be edited, because we need to remove any damaged area before we call it a mature messenger RNA. So what happens is there's a RNA processing that happens here. So the new messenger RNA is in the nucleus is not yet ready for translation, and at this stage we call this messenger RNA pre-messenger RNA. So it must go through some stages of processing before it can leave the nucleus as mature for it to be used as the messenger RNA. So this processing process has three parts. It has a stage we call splicing, it has the editing, and then it has the polyadenylation. So these processes are very important because they will mature our messenger RNA. It will make it ready to be used without having any potential for damage or potential to be modified. So here we are explaining in details what the splicing, the editing, and the polyadenylation entails. So in the splicing process of our messenger RNA, what happened is we are being told that the splicing process involves removal of introns from the messenger RNA. And by introns, we are told these are regions on our messenger RNA that do not code for proteins, while the remaining messenger RNA after that will entail purely what we call exons, and exons are the regions of the RNA that are responsible for coding for the proteins. So in short, we remove areas that don't code for any protein and remain with the areas that will code for proteins. So here's our pre-messenger RNA, or the one that is not mature, and it has exons and it has introns. Now, the intron areas, these are areas that we are being told do not code for any proteins, and so we will have to remove these. So shall remove them during the splicing. After removing them, now we will have a messenger RNA that contains only the exons. And the exons, these are portions of the RNA that will code for proteins. However, we are told we need to protect this RNA that has only the exons. And part of the process is to take it through what we call the um, five direction capping, whereby we say we add a methylated cap to the head of the messenger RNA, and this cap will protect the messenger RNA from breaking down. So in this case, it will help the ribosome to know where to bind to the messenger RNA. So this will be the binding area for our messenger RNA. So the five sequence or the 5-end capping, what happens is it will create a protection here. It's like a cap to protect it from being broken down by different enzymes or by different environments in the nucleus. And because of this, it will provide a point of binding when it's going to be translated in the ribosomes. And the last bit is polyadenylation. And in this case, we shall add a tail to the messenger RNA. So what the tail does, it will consist of a strings of adenine. So A here we are referring to the adenine. So it will add a strings of adenine bases, and these adenine bases, they are going to signal the end of our messenger RNA. So as you can see in the polyadylation process, we already added the cap in the 5-sequence capping. In that end here now we are adding the tail, which is going to be our stop signal. So it's going to initiate the stopping or ending of formation of the messenger RNA. And from there, it will now help us in exporting the messenger RNA from the nucleus. And this end is also going to help us with protecting the messenger RNA from any enzymes that can also break it down. So the polyadenylations and the capping are important for protecting our RNAs. So after we have successfully edited the messenger RNA, we have taken it through the splicing, we have done the capping, we have done the annylations. The next bit is to translate this strand of messenger RNA. So this process is where the genetic code in the messenger RNA is read to make for us the proteins. So after the messenger RNA leaves the nucleus, it will move to the ribosomes, where we will find the ribosomal RNA and the polypeptides. So the translation process happens purely in the ribosomes, whereby we can do it in the ribosomes that are floating in the cytosols or the ribosomes that are attached to our rough endoplasmic reticulum. To just bring us on board is that ribosomes are located two ways, some found in the cytoplasm while some attached to the rough endoplasmic reticulum. So what our ribosomes will do, they will read the sequence of codons in the messenger RNA, while on the other side, the transfer RNA will be bringing to us the different amino acids to the ribosome to create the polypeptides. Now, for those who are wondering what are the rRNA and the transfer RNAs, we have a whole video on types of ribonucleic acids, and you can refer to that under the video on ribonucleic acid structure, functions, and types of RNAs. We discussed the different types of RNAs, that there are 3. We have the messenger RNA, the ribosomal RNA, and the transfer RNAs. And this is where they are coming in to participate in this process. So when the messenger RNA arrives in the ribosomes, we will read that strand, and here we shall be using the ribosomal RNAs. And as we read the messenger RNA, we are able to tell which amino acids are supposed to be added. And in this case, we shall use the transfer RNA to bring these amino acids to the ribosomes in an order of their correct sequence and form the protein polypeptide. So here is a summary where we're being told in that process of making proteins, the messenger RNA will enter the ribosomes, and the ribosomal RNA will read one messenger RNA codon at a time. And then the transfer RNA will deliver the amino acids until the full protein is formed. So here is our messenger RNA strand. It's passing through our ribosomes, and our ribosomes, using the ribosomal RNA, will be reading these codons one at a time. A codon is a triplet of bases. So 3 of these bases gives us a codon or a triplet. And in the ribosomal RNA, we shall be reading one codon at a time so that we are able to compile all the given amino acids that are needed. So for example, here we are translating this, we are reading it. So as you read it, the ribosomal RNA is able to tell which amino acid is needed here. Then the transfer RNA shall bring those amino acids and allow them to create the polypeptide chain until the full protein is created. So here is a summary of our formation, that is the formation of proteins or protein synthesis. It begins with transcription, where our RNA polymerase will focus in producing for us the messenger RNA in the nucleus, and it will come from the DNA, where the DNA will temporarily unwind to help us form the messenger RNA. Now, the messenger RNA that forms here, we are being told it's not ready for use, it's not mature, so we shall call it the pre-messenger RNA. So this pre-messenger RNA will go through RNA processing or the editing process, where splicing will happen, capping will happen, and polyadenylation will also take place. And after that, we shall have our mature messenger RNA, which will go to the ribosome, and here we shall take it through the process we are calling translation. So we shall translate the codons together with the ribosomal RNA, and the transfer RNA will bring for us all the translated amino acids, and we shall form the polypeptides. So in this transcription here, what happens is we are explaining in summary, because we said the process of the process of protein synthesis has the first part, that is transcription, and the second part is translation. So in here we are going to demonstrate how the transcription process occurs, and this shows how we start from the DNA all the way to the proteins. So we have our cell here, and in the cell we have the central part that we call the nucleus. So the transcription occurs in the nucleus. So in the nucleus we have our chromosomes, and these chromosomes is a condensed state of our DNA. So we shall open up our DNA, which is rolled up or condensed through the histones. So it will detach from these histones, it will open up and explore our genes. So in the transcription stage, what happened is we are going to create an RNA copy from our DNA. So the RNA polymerase will come, help us in the unwinding or in the opening up of our DNA to start creating the RNA complementary copy. And we are saying in the RNA, we shall replace where there's an adenine, we shall do it with the uracil, the guanine will go with the cytosine, and so on, like that. And after that, when the messenger RNA has been formed, it will be complete, and it will now be ready to leave the nucleus and go to the ribosomes for the purpose of translation. So what is happening here is the editing, where we are removing any areas that are not coding for proteins, that is the introns, leaving with the extrons. So from there now we shall have our messenger RNA ready to go, and it's made up of the extrons only, or the exons. And the second part is the translation.
Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA.
So our messenger RNA leaves,
the ribosome reads the code in the messenger RNA to produce a chain made up of amino acids.
And we are saying the messenger RNA
there are 20 different types of amino acid.
Ribosomes that are
Transfer RNA molecules carry the amino acids to the ribosome.
On the rough endoplasmic reticulum.
The messenger RNA is read 3 bases at a time.
Messenger RNA
As each triplet is read, a transfer RNA delivers the corresponding amino acid.
And forming the proteins that are needed.
This
So
is
the
added
amino
to a
acids.
growing
So
chain
for
of amino
each
acids.
amino acid that is translated by the ribosomal RNA, we shall be bringing in
Once
amino
the last
acids by
amino
the
acid has been
transfer
added,
RNA
the
to
chain
give us the
folds into a
proteins
complex
for the
3D shape
patient.
to
So
form the
here we are creating
protein.
a polypeptide, organizing these proteins all the way until we have our complex structures of proteins, which are complete and can now be used as building blocks for the body in our bodies. So that marks the end of the protein synthesis. However, we are explained that during protein synthesis, some twist can happen whereby we end up with a process we call mutations. So in here, the cells are said to mutate where their genetic makeup is altered in any way. So the alteration of this DNA makeup can cause changes or modification. However, in some areas, it may cause no significant change in the cell functions. However, it can also bring modification of the cell functions and cause some physiological abnormalities. However, this will not stop any form of cell growth or multiplication. However, the client can come out with, or the person can be born with inborn errors of metabolism or defective processes like blood clotting process, or the cells can just die. And so the cell can go through cell death. So this is mutation. It just explains a change or a modification into the genetic makeup in a way that is altered and it is abnormal. So this can affect either how the functions of the cells are happening, it can modify these cell functions and cause some abnormalities physiologically. Or generally, the cell can just die through the cell death. And if you remember, this is what we call apoptosis. So this is how mutation works. It's just an alteration into our genetic makeup, producing abnormal cells or producing abnormal structure of our biological structures. That marks the end of protein synthesis. Tell me how this lesson is. Did you understand? Or do you feel some parts were not a bit clear? Just give us a comment. But also you can go ahead, share this with your friends. Also subscribe if you've not done that. And till next time, bye bye.